Hello, everybody. My name is Adriana, and I'm one of the organizers of the Teens Neuroscience Seminar Series. And I would like to welcome everybody here online. Thank you for your interest in our seminar. We now have around 60 people registered for the talk. We're still waiting for them to come online. I would like to say a few words about the seminar series. The Teens Neuroscience Seminar Series was born as an initiative of researchers in teams, which is the Transylvanian Institute of Neuroscience, and it is a research institute in Cluj-Napoca, Romania. We here at teams feel that neuroscience is still poorly represented in many countries and therefore deserves more coverage here in Romania and why not worldwide. We aim to bring neuroscience closer to those who share our passion for it, whether they work in the field or not, and inspire them to become more curious about the brain. We will hold free online seminars periodically, once or twice every month, with a wide variety of speakers from all corners of neuroscience. The format for today will be a 5-minute intro, followed by a 40-minute talk, and then a 15-minute Q&A section at the end. Please post any questions you might have in the Q&A panel of Zoom. If you hover your mouse at the bottom of your screen, it should appear as an option, and please feel free to upvote or comment on existing questions. We would like to keep this as interactive as possible, and it will be more fun if you do this, I promise. In the Q&A section, I will select a couple of interesting questions for Corette to answer, and the chat will be open for discussion as well. If you do use the chat, please select all audience from the drop-down panel so that we can all see your comments. I will now give the word to Tobias Rose, who is a professor and group leader in the Circuit Mechanisms of Behavior department at the University of Bonn. He will be our moderator for today. Thank you.
Yes, thanks a lot, Adriana. I'd also like to welcome you all for this today's seminar. It's a great pleasure for me to be around. So I have a nice connection to Cluj, especially since I'm a great big fan of the Transylvanian Neuroscience Summer School, which I hopefully will be attending in a few weeks again. So it's always a pleasure to be around there. So today, however, it's my great pleasure to introduce Corette Wierenga. So I know Corette for quite some time. So we go back quite a few years. Maybe a little bit on Corette's background. So Corette studied physics at the University of Utrecht, and she got her degree there as well, but then moved into neuroscience. And then she started studying and focusing her career on let's say, the plasticity on neuroplasticity, plasticity of neural connections. So I'd say the first big breakthrough that she made was in her original work with Gina Trigiano at Brandeis, where she studied the studied homeostatic plasticity. So homeostatic plasticity is, you probably know the general idea of Hebbian plasticity. So the idea that neurons that fire together, that show correlated activity, wire together. So there's clear correlation rules between pre and postsynaptic firing. Homeostatic plasticity is not that. So the idea there is that you need to keep a balance, an overall balance on the strength of synaptic transmission, because if you only would have this Hebbian type of plasticity, this would just lead to winner-takes-all situation where the cell would be overly excited after a while if it fires together with another cell for too much. And homeostatic plasticity is thought to in overall dampen the activity, to keep the activity of neurons at a specific and certain level. The way this works has been a bit debated, and it still is debated, but especially in the time where Corette was around in Brandeis, the locus of this plasticity, or what's more important, pre and post-synaptic plasticity, was a big debate. Same as, by the way, for Hebbian plasticity. And in her work with Gina, she figured out that, yeah, it's all not that easy. You can't really decide on clear sides of it. But I think, please correct me if I'm wrong, Corette, that you would bank on the more postsynaptic side of things here. Now, Corette then moved on to start looking into a subject that she's been following up since then. So I think I have to see, but I think in the end of the 2000s, Corette moved on to Munich, to the Max Planck Institute of Neurobiology, as it was called then, to the department of Tobias Bonhoeffer, where she started to look into inhibitory synapses, and especially how inhibitory synapses, so the transmission sites of inhibitory neurons towards other inhibitory neurons or excitatory neurons, what the rules are, how these synapses are formed, how they are structurally organized, how activity in pre and post synaptic compartments is actually changing the synapse formation. And especially she figured out how excitatory synapses are made, how excitatory neurons interact with these inhibitory synapses. So this is so-called structural plasticity. Another thing that she studied is how inhibitory synapses interact functionally at the dendrites, which are, of course, the processes that take up information of a neuron. How inhibitory synapses interact functionally with excitatory synapses using elaborate methods of precisely stimulating specific sites and recording sites where excitatory and inhibitory transmission takes place. So this was very elaborate work. And then in 2012, Corette moved on to start her own junior group, so to start her own associate professorship in Utrecht again. So she went full circle from PhD back to her independent work. She did this with one of these nice Dutch, very competitive grants, which I think was Vidi or Veni or Vici. I always mix them up, but it's great to have them. And Corette continued to study the interplay of inhibition and excitation, which has been a big theme, which is good because turns out that's a really important subject. So the original idea of inhibition is just something that just dampens the whole networks, that maybe is a blanket thing that keeps everything roughly in check, is wrong. Turns out that inhibition is much more center stage in neuronal networks. Effectively, one could say to some extent it's at the driver's seat of network states, and the plethora of inhibitory subtypes, the increasingly large variety of distinct connectivity rules between inhibitory cells and excitatory cells also warrants a lot of further research into that. And I'm really looking forward into what Corette is going to talk about today, and I think it will be along this line. So please, without further ado, I'd like to welcome Corette to the stage / switching on the video. And I'm really looking forward towards your presentation. And yes, I invite questions, which I will be forwarding after the talk together with Adriana. Thanks a lot, everybody. Welcome, Corette.
Thank you, Tobias. Thank you for this very kind introduction. You gave away basically all my secrets already, so I don't have to give the talk anymore.
Oh, there's this 91 prize you got in Russia. We should talk about it.
Yeah, that's a long time ago. No, yeah. So thank you, and I'll just basically start with sharing my screen if I can find it. This one. Yeah. Let's see. Is this now the right one?
We see you. No, it's right now. It's good.
Now it's a good one, right? You see not the.
All good.
Oh, good. Yes. So I don't. Did you mention? So I just moved basically from Utrecht to the Radboud University to Nijmegen.
I forgot this. Congratulations.
Doesn't matter, but
Corette is now full professor in Nijmegen, which is even closer to us.
Doesn't matter, but I noticed this thing like, oh yeah, I should mention that I'm now in Nijmegen, and I'm sort of halfway there. We are. The laser is packed today, and it will be unpacked on Monday. This is basically the status of my lab. So I'm a little bit off here, but anyway. So today, what I we'll talk about is exactly what Tobias already introduced, the balance between, or the coordination, I rather say, between excitatory and inhibitory synapses within dendrites. And just because I'm not 100% sure what the background is of the audience, I begin at a very, I would say, very basic level. What happens in your brain when you perceive the world around you? Sensory inputs will activate some cells, and those will activate other cells. And basically, this is how information gets processed by groups of cells, little samples of cells in your brain. But if you then pick out basically one of these cells and you zoom in, you would see that it receives thousands of inputs from other cells, and basically it gets two types of inputs: excitatory and inhibitory synaptic inputs. And excitatory synapses give a small positive signal, and the inhibitory synapses give a small negative signal. And as Tobias mentioned, like basically the only way this cell could do its work properly, process the information, is if it has a sort of a coordinate set of excitatory and inhibitory inputs, so that sometimes it would respond to an input that it should respond to, but it should ignore others. It should not always respond. And basically, for a long time, it was thought that these inhibitory synapses are just there to basically sort of dampen too much excitation, basically a simple counterbalance of the excitatory signals. But basically, it's now becoming very much clear that these inhibitory synapses are actually doing much more sophisticated things. And one thing that you can appreciate is that basically so there's different types of inhibitory synapses, different types of inhibitory cells in networks, and they can be active under different circumstances. And in this way, they can basically decide or determine which cells are active at which time, and which inputs they are receiving, or which inputs they are processing. And this is a little bit, I try to put that here. It's basically the same network, let's say, in the visual cortex processes visual information, but under different circumstances, the information is processed slightly differently in that network. You can imagine yourself when you're in the supermarket and you look at all the groceries that they have. You see different things when you're hungry or when you just have eaten, or you also see different things when you're in love or when you're really pissed off. So this is really, and this behavioral context that's very often provided by neuromodulatory inputs to inhibitory cells in the brain. Now, okay, that's basically on a systems level. So how does it work basically on a cellular level? And for that, you should know, and probably many of you know this, that when you have two synapses that are very similar, the location very much matters, right? This synapse is very close to the soma, has a strong influence on the soma, while this synapse, the signal from the synapse needs to travel all the way through the dendrite, which will attenuate the signal, and so the signal will be much weaker in the soma. But the brain has overcome this by very often what happens is that synaptic inputs that carry sort of similar information are clustered together within the dendrite. And then in that way, they can basically cooperate and have a strong impact on the soma. Now, if you then look in the dendrite of cells, there is like similar multiple clusters of inputs in the dendrite, and then again, it's not only excitatory inputs, but there's also inhibitory inputs. And we know from work by ourselves, but also from others, that these inhibitory synapses, they have mostly effective on excitatory synapses close by. So Okay, as I said, that is the task of this red cell that might be different under different circumstances, if you're hungry or not hungry, or in love or whatever. And so then the behavioral context that, as I said, is very often provided by neuromodulatory inputs to the inhibitory cells. And these neuromodulatory inputs, they come from different brain areas or long-range inputs. And basically what then happens is that under these circumstances, when these two inhibitories, I'm not sure if you see my my mouse,
yes.
But when these, yeah, you see, okay, good. So when these two cells are active, these two inhibitory cells are active, basically the inputs from channel B and C will be inhibited, and this red cell will only process information from channel A. In other behavioral contexts, maybe this cell will be active and channel A will be shut down, and the cell will only process information from channel B and C. And then, of course, you can make things more complicated, that interneurons inhibit each other, but this is sort of the general idea. And so basically what the take-home message of this idea is that these inhibitory inputs into the dendrites of cells, they are able to gate excitatory synaptic inputs. And what is really important here is where they are located. Right, so an inhibitory synapse should be close to the excitatory synapses that it wants to be gating, and that this that's the location of inhibitory synapses that really matters is also clear if you just look at the anatomy of or morphology of some of the inhibitory cells in the brain. For instance, here's a CCK interneuron in the hippocampus, and you see that it makes a lot of inhibitory synapses really sort of aligned with the Sheffield collaterals that are basically the excitatory synapses from the CA3 to the CA1. So it really has its inhibitory synapse aligned with a specific type of excitatory input. Now, as I said, so I'm very interested in trying to figure out how are inhibitory synapses formed, who decides when and where they are formed, and basically what are the molecular mechanisms there, and how are these regulated in the normal brain and maybe dysregulated in diseased brain. Now, the way that we study these inhibitory synapses is by using a transgenic mice. Transgenic mice get 65 GFP mice. Those have GFP expressed in some inhibitory cells, and it's basically the inhibitory cells that target dendrites. And so you have this beautiful, if you make here a slice of the hippocampus, you see that there's beautifully labeled green cells. Those are all inhibitory cells. And then what we do is that we fill one or a few pyramidal cells, so excitatory cells, with a red dye, for instance, via patch pipettes. And then when we zoom in, you can basically see how these green axons, which we know that are inhibitory axons, make now synapses to this red dendrite. And the way we make these beautiful images is with two-photon microscopy, which has the big advantage that everything is alive, so you can follow things over time. And this is exactly what we do very often. We take images every, in this case, every half an hour. We now do it usually a little bit faster, every 10 minutes. But we basically follow structural changes over time in these slices. And in this case, this is basically from the first publication that I did with Tobias Bonhoeffer. Here we have a red dendrite and a green axon, and this green axon at some point in time decides to make a new inhibitory synapse onto this red dendrite. And in this case, I didn't do anything. I was just sitting and watching. But today I will tell you that we now have an idea of the mechanisms that can be at play that decides when and where these inhibitory synapses are formed. And basically, these experiments that I'm telling you, that I will tell you about, are based on the idea that for an optimal system has some coordination between excitation and inhibition. To say it simple, it's a balance between excitation and inhibition. And in these experiments, what we will do is we will disturb this balance and see if there are mechanisms to restore it. And the way we're going to disturb the balance between excitation and inhibition is that we make excitation stronger. And we do that by using glutamate uncaging. And I just, as I'm not sure if all of you know this technique, I explained it here. And basically in glutamate uncaging, you use an inactive form of glutamate, glutamate in a cage glutamate, and you have that in the slice. So by itself, it doesn't do anything. But if you shine a laser at a certain spot, at that spot, the cage will be broken and the glutamate will be free. And basically, basically where you put your two-photon laser, that's where you have a little local sniff of glutamate. And so if you do that in front of a spine where there's an excitatory synapse, basically every time you uncage glutamate, you can measure in this red cell, you can measure a small EPSC, a small excitatory postsynaptic current, and it's very reliable, if you can see. And then what you can do is not just give one pulse, but you can give a little train of pulses. And with that train of pulses, you can basically induce LTP, you can induce potentiation in this specific spine. You see that this one has grown while the others around it have not changed much. So, and this is just to show you that indeed with our protocol that we use, we can indeed reliably induce spine growth in our dendrites. So this is what we do, right? We have a dendrite where there's excitatory and inhibitory synapses. We specifically look for basically a place, a location in the dendrite where there is an axon, an inhibitory axon crossing the dendrite, but there is no inhibitory synapse yet. And then we do the glutamate uncaging so that we make excitation stronger here locally. And by this, we will disturb the balance, the local balance between excitation and inhibition. And we will basically ask, so is there now something that will restore the balance? For instance, by making a new inhibitory synapse. Here's then the actual experiment. Here's the dendrite. Here's the 4 spines that will be stimulated to induce the potentiation. And basically, we start by just taking some images before baseline imaging. Now we do the uncaging, and you should focus here on the middle. First, nothing seems to happen, but then at some point, like here, now you see that something seems to have been growing here. And I think you can see it better if I show you this one. It's basically the before and after picture. So this little piece of axon is what you see here, this little piece of axon, and you see that over time, there's a clear blob growing, and we call that a bouton. That's basically what you call a presynaptic terminal. So basically, what you see here is that, like, after you trigger potentiation of excitation, the inhibitory axon reacts to it by making a new inhibitory bouton. And basically, the idea is that this will then restore the coordination between excitation and inhibition locally. Now, this is the how do you say it? The quantification of this one experiment. Here is quantification of multiple experiments. And the way you should read this is time on the x-axis, and on the y-axis are all the experiments that we did. And when it stays blue, like here in the upper ones, that means nothing really changes. But when you see something red or green, red or yellow, that means a new bouton has grown. And so when we don't stimulate any spines, you see that in most cases, nothing really happens. But then when we start stimulating spines, then you see that now there's specifically more bouton growth on the inhibitory axon. You also see that it's not happening every time, right? Not all inhibitory axons respond to it, but there is a clear increase in the number of inhibitory boutons that are growing. Now, I won't bore you with all the details, but basically what we found is that the dendrite sends out a molecular signal to the inhibitory axon to basically ask for a new inhibitory synapse. What we found is that this signal, this molecular signal, is endocannabinoids. And this is how we think it's the mechanism then goes by the glutamate engaging, by activating glutamate receptors. There's a calcium increase in the dendrite, and this through a complicated pathway will activate the enzyme DAG lipase, and that is the enzyme that can produce endocannabinoids, in this case 2-AG. And this 2-AG, the endocannabinoid, can then bind to CB1 receptors, endocannabinoid receptors on the inhibitory axon, and this will trigger bouton growth. Now, just to show you that that we have also some evidence for that, basically what we did is we use a specific blocker for CB1 receptors, and then what we see is that we can still happily induce spine potentiation, but basically no bouton ever grows in when we have the CB1 receptor blocked. The other, this is then the quantification. The other thing that we did is that we have a specific blocker for the enzyme, and actually we put that blocker in the pipette. So, and also that completely blocks boutons growth. So that means that really it is this red cell that is producing, basically the postsynaptic cell that receives the stimulation, the excitatory stimulation, that cell will start producing the 2-AG that is required to trigger the inhibitory bouton formation. So that means that the dendrite can direct when and where a new inhibitory synapse needs to be formed, exactly at the location where excitatory synapses are active. Good, so that's one thing. And then we wanted to understand a little bit better how activation of these CB1 receptors can actually trigger inhibitory synapse formation. Because from literature, we know that CB1 receptors and axons, those are important during development for axon guidance. They are also able to modulate synaptic release in the adult brain. But this was actually the first time that CB1 receptors were linked to synapse formation. And so then what we did to address this, we basically zoomed out and only focused on the inhibitory axons, the GFP labeled inhibitory axons. And we acquired basically images of these axons every 10 minutes and follow their boutons. Basically what you can then do is really zoom in and follow individual boutons over time, and you can see basically when new boutons are formed over time. And what we did is then we take a baseline, then we wash in Win, in this case, that's an activator of the CB1 receptor, it's a specific agonist, and then we continue imaging to see if activation of CB1 receptors can indeed trigger bouton formation. Now, what we realized when we did these experiments is that not all inhibitory axons have the CB1 receptor. In fact, it's about 50% in our hippocampal slices. So what we can do, we have a very good antibody for the CB1 receptors. And basically what we can do is we label all we do it here. So we do the two-photon imaging, then we fix the slice and find the same axons back in the confocal. And then we stain for CB1 receptors, and then basically we can identify what this axon is completely covered in CB1 receptors, while this other green axon does not have any CB1. So we can basically make the distinction between CB1 receptor, CB1 positive and CB1 negative axons post hoc. So what we then So we did the experiment, and basically what we see is that when we wash in this wind, we activate CB1 receptors, we can indeed induce inhibitory Bouton formation, but only in the axons that have the CB1 receptors. In the other axons, which are in the same slice, which have the same treatment, they don't respond to the CB1 activation. And we think that actually this may very well explain why we don't always see formation of inhibitory Boutons after we do the glutamate uncaging, because only half of the green axons actually are responsive to the endocannabinoids. One thing that I wanted to mention here is that basically these new Boutons that we are seeing, they are not yet full-blown synapses yet. So we know from, we've been working on inhibitory synapse formation for a couple of years already, and we know that it's a very dynamic process which includes multiple molecular steps. And so basically the first step is that basically an empty axon will make a new Bouton, but that new Bouton is first unstable and immature, and only after it's stabilized, only then it really forms an inhibitory synapse. But what we see here in the after the CB1 receptor activation is that we really only made this new sort of immature Bouton, and we know that it takes about 2 to 3 hours or so before these newly formed Boutons really recruit presynaptic VGAT and postsynaptic Gefrin. They're basically markers of an inhibitory synapse. So that's just to tell you that it's not making a new synapse at the spot, but it's basically making the first step towards a new synapse. Anyway, so I said we wanted to understand how activation of CB1 receptors can actually mediate synapse formation. So it's not new that dendrites produce endocannabinoids. That is well known, but basically the most well-known function of these endocannabinoids is that it has as a retrograde messenger, it basically suppresses the release of presynaptic neurotransmitter, in most cases in GABA. Now that is of course opposite more or less to what we see in our experiments, where it doesn't suppress the release of GABA, but it really triggers the formation of a completely new Bouton. But luckily, because it's such a well-known retrograde messenger, also the mechanism is very well described. And so we can use basically that mechanism and to see if in our case, CB1 receptors use the same mechanism. Now, we know that for the suppressing of the GABA release, the CB1 receptors, they are linked to GI proteins, protein signaling, and GI protein signaling then suppresses release. So in our experiments, what we do is we use a specific blocker to block GI signaling, and then check. And then check if we can still induce inhibitory Bouton formation. And actually, this was quite surprising that when we block GI signaling, CB1 receptor activation still has a strong effect on Bouton formation. In fact, it seems only to become more pronounced. So that really means that in this case, CB1 receptors use a very sort of unconventional signaling mechanism. And so for a while we were a little bit puzzled, but then we were happy to find more recent papers where basically a new form of plasticity is described that shows that CB1 receptor activation can also induce potentiation of synapses. And it was suggested that maybe this happens or it requires presynaptic PKA activity. And so if you think about it, how can this be? CB1 receptors are normally GI protein coupled receptors, and GI proteins, their job is basically to inhibit the formation of the production of cyclic AMP. So they inhibit AC, and therefore the production of cyclic AMP is stalled. And cyclic AMP is then normally activating PKA. So this is basically how CB1 receptors normally act. But there are some descriptions in the literature that shows that CB1 receptors, and actually maybe all G protein coupled receptors, they can also switch basically to which G protein they are coupling to. And so under certain circumstances and in some cells, CB1 receptors can actually also couple to the GS proteins. And GS proteins, the S stands for stimulating, has the opposite effect. So GS proteins activate adenyl cyclase. It basically promotes production of cyclic AMP and therefore promotes activation of PKA. Now, to test if maybe this pathway is involved in formation of inhibitory synapses, we decided to use a specific blocker for PKA. And basically what we find is that if we block PKA, then we completely block the effect of, we can basically completely block inhibitory boson formation through CB1 receptor activation. And what is even more interesting is that if we artificially stimulate cyclic AMP production by putting forskolin for 5 minutes in the bath, we see that we actually have an increase of this boson formation. So basically, this really suggests that enhancing cyclic AMP levels, that is sufficient to trigger a boson formation. The time, yeah. Yeah, and so I mean, what this basically shows is that cyclic AMP is important, but it's not a very specific experiment, because basically if we put forskolin in the bath, we basically trigger cyclic AMP production in all cells in the entire slice. So to make it more specific, what we use the DREAD, which is a designer receptor exclusively activated by designer drugs. Basically, what it means is that it is a sort of modified G protein coupled receptor that is only activated by a specific agonist that in normal slices doesn't do anything. Only in the cells that express this GS DREAD, it will start. It will promote cyclic AMP production. And so basically, what we do, we make use of two viruses to have GFP labeled inhibitory cells, and to target inhibitory cells, we now make use of a different mouse line. It's VGAT-CRE. So basically only inhibitory cells, they will express VGAT and therefore they will express CRE. And so they only in those cells you have expression of the virus. So we have inhibitory cells that express GFP, and so we can follow them under the 2-photon. And we have inhibitory cells that express the GS DREAD. And sometimes they overlap and sometimes not. And then what is nice is that, again, we can activate this GS DREAD, so we can basically stimulate cyclic AMP production, but only in the purple cells. And basically, then we can look in the GFP labeled axons, and we can see if, in this case, when you activate GS DREADs, we see that it triggers boson formation in cells that have the GS DREAD, but not in the ones that do not have GS DREADs. So basically, this demonstrates that really an increase in the cyclic AMP levels, that is sufficient to trigger the formation of new bosons in inhibitory axons. So, yeah, I just wanted to briefly summarize what I showed you today. First of all, I showed you that the dendrites of CA1 cells, they are active signaling compartments. So when a cluster of excitatory impulse is active, or it's overactive maybe, then the dendrites start producing an endocannabinoid signal very locally, and this will trigger the formation of inhibitory bouton growth really at that dendrite. And such a local feedback mechanism that assures that there is really local inhibitory control over clustered excitatory inputs, which I think is an important way of really having local coordination between excitation and inhibition. And as I said before, this oh yeah, sorry. And then the next point is that, of course, I showed you that activation of CB1 receptors on these inhibitory axons, they can trigger the formation of new boutons. And this is not via the conventional GI signaling, but this seems to be mediated by an increase in cyclic AMP levels and PKA activity. Right, and then again, I want to stress that these new boutons that are formed, those are not full synapses yet. It's just the first step, and we actually previously have shown that this second step, bouton stabilization, that actually requires activity. So this actually suggests a very nice two-step mechanism in which first a bouton is triggered, a new bouton is triggered at the dendrite on a location where they are most needed. And then there is the second step, and then that decides if these boutons are stabilized or not, and that is only when these inhibitory axons are active. And I think that such a two-step model can very nicely explain how feed-forward inhibitory synapses get very nicely aligned with excitatory inputs, basically as I showed you in this example. And for this, it's important to mention that these CCK cells that do this perfect alignment, they have very nice CB1 receptor labeling on their axons. And the other thing that I wanted to, basically the other point that I wanted to make is that something on the cyclic AMP signaling. So as I explained, it's pretty uncommon for the CB1 receptors to signal via an increase of cyclic AMP because they're usually coupled to the GI proteins. But of course, there are many other GS protein coupled receptors in the brain that are just also normally coupled to GS. And it turns out that some of these have already been linked to bouton formation. And probably the best example comes from this work from Eric Kandel and Greg Bailey. So they already described 30 years ago that memory formation in the aplysia, the sea snail, I think it is, basically memory formation in the aplysia involves the rapid formation of new boutons, and they showed that this is mediated by an increase in cyclic AMP, in this case via the serotonin receptors. But also in other model organisms, people have made the link between cyclic AMP signaling in the axon and presynaptic bouton formation. And actually, this is something that we're following up at this moment. We're trying to use a PKA sensor to basically directly measure PKA signals in dendrites and axons, and try to see if we can really are able to pick up local increases in cyclic AMP or PKA, and to link this to a local bouton formation. So basically what we suggested, that what we found now for the CB1 receptors may be much more common in the brain. It basically suggests that axonal G protein coupled receptors may be not only there to modulate synaptic transmission, but really they may play a role, a crucial role in context specific or axon specific synapse formation. So with that, finally, I'd like to thank all my amazing lab members. They make working lab a lot of fun. And I'd like to thank you for your attention, and I'd be happy to answer some questions if you have. Thank you. I'll stop sharing, I guess.
Thank you very much, Corette. That was lovely. It was very, very interesting. So thank you for starting. We do have a couple of questions now. So I will start with the most upvoted one, if everybody is ready. So from Razvan, what does the inhibitory axon need to start the process? To be physically touching the dendrite? Or is there a signal from the dendrite which makes the inhibitory axon move until it touches the dendrite and then starts the formation of the synapse?
Yeah, that's an interesting thing. We think it's somehow already attached. So of course, under the two-photon, everything is alive, and you see that these axons and dendrites, when they're crossing, sometimes when things are moving, you see that they are moving together. So they seem to be already some kind of an adhesion. Basically, before you see any synapse or any bouton. So we think there might already be sort of a flagpole, like here's a potential synapse, and it's only basically being made when it's needed. And we see that very often these presynaptic terminals, they are very dynamic. You can easily see them being formed there within a couple of minutes, but they can also disappear within a couple of minutes. And so when we do this time lapse imaging, we see them basically coming and going all the time. But always on a specific spot. It's not that sometimes it's here and sometimes it's 2 microns away from it. No, it's always on that. So it seems that really there's is some kind of a marker there.
There's a seed of a synapse there.
Yeah. Or well, I think the way I see it is that there's probably some adhesion molecules already on both sides of the connection that marks a potential synapse. Yeah.
And would this be a necessary condition? Do you think there can be this effect if they are not physically touching?
Well, there's also stories from basically cultured cells where they put basically a cell on a culture dish, and then it grows out an axon. And sometimes when the cultures are very sparse, axons can be very long, and you can see that they also form boutons, only there's basically no postsynaptic cell present. So I think, and there you also see this dynamic behavior. So I think it's not necessary. I think you can, in principle, make boutons everywhere along the axon and also pretty rapidly. It's just that I think that when you have a connection already, an axon dendrite crossing, that basically you sort of have a higher chance, or basically, you, as I said, it's sort of the flag. Here's a potential synapse. If you need it, you can build it right now. Yeah, but we don't know what that molecule would be. I have some ideas, but of course, then there's no good antibodies versus those things. Yeah, but that would be really interesting to see that that very first coupling, basically. Yeah.
Yes. There's a question that is from an anonymous visitor, which directly ties into exactly that question. And I guess you've heard this many times before. So it's effectively the question, how on earth do you know it's a synapse and not just a swelling effectively?
Yeah,
right?
yeah. So that we don't, basically, because under the two-photon, we just see basically a structural blob. Basically, but what we can do, what I rushed over quite quickly, but what we can do, we can basically fix the slice right after the live imaging, and then stain for pre or post-synaptic markers, and then we can basically, we know how long the bouton was alive before we fixed the slice, and we can basically correlate if there is pre or post-synaptic marker present to how long that bouton was alive. And then we see that when it's just there, it's usually empty. And so it doesn't have any pre or post synaptic marker. So you could say it's not a synapse yet. And then over time, it acquires usually first the presynaptic marker, and then like an hour later or so also the postsynaptic marker. But that doesn't mean that every blob that we see is forming actually a synapse. But on average, we know that it takes such a, and we know also from work actually in Drosophila, where they did it much sort of on a closer time scale. We know that there also boutons are first formed, and they call them ghost boutons, because it's basically just an empty blob. And then they're very rapidly filled with synaptic vesicles. And then only then basically the true synapse is being formed. So the active zone will then be formed and the postsynaptic site will be formed. And I assume that also in these inhibitory synapses, it forms basically a similar process. And we know already some of these steps. And we think that this cyclic AMP dependent endocannabinoid dependent trick is really only the first step, basically making a blob and filling it with synaptic vesicles, but it's not triggering making a postsynaptic site, basically. And we think that their activity is needed. Yeah.
Yes, great, thanks.
Thank you very much. I had a small question of my own regarding this matter. I was wondering whether you see a postsynaptic effect in these immature blobs that you see. Is it a discrete transition from a mature to from an immature to a mature synapse, or is it really gradually influencing the postsynaptic side?
Yeah, so I think 10 or 20 years ago, people have tried to sort of figure that out. And there are, on the presynaptic side, there are basically in the axons, there are vesicles with sort of ready to go active zones. And basically when a new presynaptic terminal is being made, synaptic vesicles are sort of recruited from neighboring boutons, but then you need a few of these active zone vesicles that sort of, how do you say it, like sort of make this prefab active zone. And there you can really see that you have from 0 to 1 and to 2 basically active zone particles. On the postsynaptic side, it's really a sort of a gradual process. So the slowly Gefferin in my case is sort of recruited to a synapse. So I think it's just the way I see it is that the presynaptic terminal is very rapidly formed, and that will send out not only by neurotransmitter release, but maybe also through another adhesion molecule, sends out a signal to the postsynaptic site to now start making the postsynaptic specialization. Yeah, but yeah, I mean, we haven't shown that basically. It's basically what I'm telling you now is basically my idea.
Yeah, understand.
Yeah.
We'll go back to another question that we have. Are these receptors present in the cortical neurons? Excitatory inhibitory balance is largely related to the cortex. So what can be the mechanism to maintain the brain at a critical point? They also say, thank you, it was an amazing talk.
Thank you. Yes, so these CB1 receptors are basically present everywhere, and they are really highly expressed in a subset of interneurons. So I showed you 50% of the green interneurons in our slices, but I think in overall percentage, it's maybe 20 or 30% of interneurons in the brain have them. And what is interesting about it is that they're very much localized on the axon. So when you basically look at the stain for CB1 receptors, you don't see any cell bodies, because cell bodies hardly have any CB1 receptors on them. But you see very nicely, you can follow all the axons and the boutons. And so it really looks like they have a specific function there. And in my view, the specific function is synapse formation, and the way we show it. But yeah, so it's in the cortex, in the hippocampus, it's everywhere. And we think that these cells are really probably the function of these cells is really to basically coordinate the inhibitory synapses on close to where excitatory synapses and where the endocannabinoids are being formed. Yeah.
Okay, thank you.
All right, if I may, I'm squeezing in a question of my own. So to close the link between CB1 receptor activation, cyclic AMP increase, PKA activity, and local synapse formation. I mean, what you're doing, starting to do, obviously, is looking at PKA, local signaling, and I think that's a crucial point to really go down to see when these local signals actually occur.
Yeah.
And so I mean, there have been quite some developments in recording GPCR-based signals. So Lin Tian's lab, is there a CB1 circular permuted GFP on the menu?
No.
Totally doable. Why not? I mean, talk to them, because I mean it's exactly the same principle for all these G protein coupled receptors. They can not all be done that the circular permutation trick has been solved. That would be awesome.
Yeah, that would be cool. But I think the nice thing about PKA or cyclic AMP imaging is that it's more general, right? It's not specific for CB1, because actually we have indications in our lab that it's really not specific for CB1. We can, when we do, we know that many of these inhibitory cells in the hippocampus also express D1 receptors, the dopamine receptor. And when we do the same trick, we give 5 minutes of agonist of the D1. We can also induce new inhibitory boutons. So it really seems that it's basically a general trick, and you can have you can do this local EI balance for endocannabinoids, but maybe you can also have a modulatory signal from, I don't know, dopamine or maybe serotonin, and that can do also a similar trick. And I think that is, to me, that's very interesting, because then you can really make inhibitory synapse formation really sort of context dependent, and very rapidly. You can rapidly make new synapses, or maybe also remove them. And that's actually what we know from studies that, for instance, when you have a mouse learning a new trick, basically a motor trick, and you look in the motor cortex, you see that what happens very rapidly is that inhibitory synapses go away, spines rearrange, and then you have basically an EI balance. So you have this constant, it needs to be constantly rearranged, and we think that these neuromodulatory signals might play a very important role here. And this is really something that we want to study further in the future. Yeah.
I mean, of course, you're making the claim that this is super local, right? And I mean, so cyclic AMP is a diffusible factor. So it's making the point that this can actually be local, would potentially be important in the future.
Yeah.
I mean, there have been developments in the light activated adenylyl cyclase field. There's now membrane targeted PAC, so Georg Nagel's tool. So Thomas Oertner and Christine G are working with that a lot. Of course, I don't know, this would be cool if you could have a local activation of a stretch of axon that could show that you can get boutons there.
That is certainly, those are the type of things that we're thinking about. Yeah.
Very
but we're
cool.
also doing on the postsynaptic side, we want to know what decides when and where endocannabinoids are produced, because I think that is also what is very local. And I mean, if you read the literature, yes, it's activity dependent, but when and where exactly it's produced, that's basically not very clear. And we actually tried, there is now a very nice grab sensor for endocannabinoids, but it's not nice enough that you can see basically individual dendrites. That would be our dream, right? You have one dendrite and you do this 4 spines and then you see ploop, you see endocannabinoid making. But yeah, that didn't work. You see, when you do a lot of depolarization, yes, you see that the sensor senses endocannabinoids. So, but it's just not sensitive enough that we can use it for this. But yeah, hopefully in the future.
Yes, that's what I say about tools these days a lot.
Yes.
So there's glia as well, right? In that context, in the cannabinoid signaling. Yeah,
absolutely. Yeah. The thing is that the fact that we basically see responses only in the CB1 positive axons and not in the CB1 negative axons,
Right.
makes it to us that it's really the CB1 receptors on the axons that are active. And it's really the same axons are in the same slice, they're everywhere, but they don't respond. So it seems that maybe glia have a role here, I don't know, but not in like the CB1 receptors. It's really on the axons that are involved here. Yeah.
I mean, I could continue, but we should probably have the opportunity for somebody else. You want to ask a question, Adriana?
Yeah, should I read the next? There's an anonymous attendee that asks, how do you deal with variability of GFP expression?
Yeah, there is not too much variability in our slices. And also it doesn't matter because we basically look over time. So you have stronger axons and weaker axons, or like stronger or weaker intensities. But yeah, we just follow them over time. We generally, like for our analysis, if they're too weak, we just basically leave them out of our analysis, because then it might be hard to basically identify boutons. But other than that, no, it's basically not big of a deal, I think.
You take a baseline and then you just reference everything to that baseline.
Yeah, exactly.
Get rid of all.
Yeah.
I understand. Tobias, you want to read another?
I can do that. So let's do the question of Razvan. Does every branch have at least a single inhibitory synapse at its base? So I assume does each dendritic branch have a gating synapse?
Yeah, it's not so much that it should be on the base. Actually, just it should be anywhere. But yeah, there are studies that basically you have a very nice coordination between excitatory and inhibitory synapses within dendritic branches. So there's EM studies people who have really painstakingly going through all the dendritic branches of cells and really looked at what is the excitatory and how many excitatory and how many inhibitory. And yes, that is very strongly correlated. And actually these original experiment was based actually on an EM study from Kirsten Harris, where she showed that you always have this nice correlation between excitatory and inhibitory synapse along a dendrite. And then she did LTP induction locally, and then she sees that it first is basically off because excitation becomes too strong, but then she showed that 2 hours later inhibition is sort of how do you say it? Yeah, compensating for it, and that made us think like, hey, okay, then there should be some kind of, and this is actually how this whole thing started. Yeah, but yeah, so that is very well described actually.
Thank you. So we have one more question left here. What else is required in order for a synapse to be fully mature? I think they refer to the fact that you mentioned those immature synapses, and then what is required to make the transition from an immature to a mature? Yeah, so we know that the first step is then to make basically a new bouton, an immature bouton, and the next step is to stabilize that bouton. And we have a paper that shows that semaphore D signaling is important for that, and that basically requires really a remodeling of the actin skeleton in the presynaptic terminal. And then there's papers from other people that show that on the postsynaptic side you need to basically build gephyrin, that's the scaffold molecule, and you have to have receptors. And there is an interesting link between calcium signaling and gephyrin. And so it really depends on calcineurin and chem kinase 2, basically the same things that also are important for excitatory synapses. Those are also important for building the postsynaptic density of the inhibitory synapse. So it's really different molecules are important at different steps.
which sort of gives to me a sort of a checkpoint system, right? You do the one thing, so you make inhibitory synapses, but maybe everywhere, but only the ones that are actually also activated, only those you keep, right? That seems to make sense to me.
Okay, but this pathway is activated by CB1 only, so you don't need other
No, the CB1 is really only activating the first
step. Okay.
Yeah. No, and actually, so what we try to do is because we saw that these new photons that we are making, they are unstable, right? Unstable. They sometimes go away also during the. So we then wanted to make full-blown inhibitory synapses, and so what we did 3 times stimulation, not with the glutamate and caging, but basically just activation of the CB1 receptor 3 times, and then looking basically 24 hours later. And then indeed we see that we have an increase in mini iPSC frequency. So we thought, okay, so we have really made new inhibitory synapses. But then when we do the staining, we see that we have actually more VGAT puncta. But these VGAT puncta, the new ones, are not linked yet to Gefrin, because of course we did not increase the activity in the slice, right? We just only pushed the CB1 receptors, but there's no sort of follow-up. And that is actually, I thought that was really interesting, that we make sort of immature synapses waiting to be stabilized, and then nothing happens, so then they stay like that. Yeah.
There's no handshaking.
Yeah, exactly.
Yeah, understand. I just have a very short question of my own. Does the age of the animal have any influence here, or?
No, we use slices from P7 mice, okay. And then we put them in culture for, well, diff 10 to diff 20. And so in that range, we looked like, does it more happen in diff 10 or diff 20? We don't see any correlation, so I don't think it matters. And we know that endocannabinoid signaling is present already very early on, and these CB1 receptors are there from basically from the get-go. So I don't think there's much.
This is not developmental at all. It's
although if you read the literature, there is developmental plasticity, endocannabinoid based developmental plasticity. So it might be that that in certain regions it might be developmental regulated, but in our small, of course it's a very small range, diff 10 to diff 20, there we don't see any big effect of the age.
Thank you.
Yeah, a small question from my side. This strange binary expression of endocannabinoid receptors in the same culture is of course very interesting. Has this been shown in vivo as well? Because this could
binary.
You said that some axons do have CB1 receptors, some axons totally don't.
That is totally the same in vivo. Yeah. It's just some interneurons have expressed CB1 and some don't.
So is this a type question, or is this a branch question, or is this?
No, it's a type question. So
I see.
Cell has it or not?
I see. Okay.
Yeah. And for instance, PV cells don't have much CB1.
Right.
But all CCK cells have plenty of CB1. So it's it's a type.
We have received one more question.
Okay, sure.
It seems like they won't let us go.
Yeah, sure.
Is it possible to link CB1 receptors to the inhibitory interneurons of sensory systems, provided that deactivation of these inhibitory interneurons results in reduction of efficiency of the sensory system?
Yeah, so we done all these experiments only in hippocampus, but my guess would be that you have similar, basically that a similar effect would be happening in sensory systems, because we know that in sensory systems there are CB1 positive axons, and these pyramidal cells can make endocannabinoids. So there's no reason why it would not work like that. So I think I don't fully understand the part about
he says that deactivation of these inhibitory interneurons results in reduction of efficiency of the sensory system. So I guess
Yeah, so you,
the presence of these inhibitory neurons is useful for the actual function of the sensory system.
Yeah, so the way I think of this is that it's a really good system to make this sort of alignment of feed-forward inhibition with an excitatory input. But it should, but like for different, of course, there's different types of inhibition. And there's also feedback inhibition. There's also inhibition of inhibition, basically disinhibition. And I think this is how I see it, that like some cells have CB1 receptors and some don't, because some cells, they, for instance, PV cells, they make connections on the soma of their target cells, and this may not be guided by CB1 receptors. It really may be just an activity dependent thing, right? And other cells need to make synapses onto other inhibitory synapses, which also needs to be guided by a different molecular mechanism. It's not to say that this CB1 dependent mechanism is everywhere and is always. I think it's just one way of making a very specific very specific formation of inhibitory synapses, basically by aligning with the excitatory inputs. But other inhibitory synapses might be organized in a different way. And I think to have a full working network, you need, of course, all different kinds. Yeah. But yeah, I think this also should work in the sensory cortex. But yeah, I hope people start looking at it, and hopefully they find interesting things.
Thank you very much, Corette. I don't know if this is okay with Tobias. We can wrap it up.
Absolutely.
It was great to have you both here, and thank you very much for the interaction and for the questions. And the talk was amazing, as many people have mentioned. Thank you very much for being here, for accepting our invitation, and we do wait for you again. Also, Tobias will already started scheduling your talk in this seminar series, so hopefully we're gonna have even more fun then. And for everybody else, please join our newsletter if this was interesting. We have a link on the poster, and also where on the website where you registered for the talk, you can subscribe to the newsletter to receive news. I think the next talk will be maybe after the summer, because we have some events scheduled, like Tobias has mentioned, the TENS school, the Transylvanian Neuroscience Summer School. But we will keep you posted, and please join us again. Thank you very much for your attention and for your participation.
Thank you very much for inviting me. It was really nice to do. Good.
It was great fun. Nice seeing you, Corette. And
yes, absolutely.
I see some of you soon.
Yes.
Hopefully you too.
Yeah.
Bye.
Bye.
Bye-bye.